Thanks, Emily, for that really kind introduction. So I chose this title partly because Age of Ultron didn't really fit well with what I wanted to talk about, but mostly to describe what an exciting time it is to be in psychiatry. I really feel with advances in genetics, imaging, evidence-based treatments, we are really close to being able to treat patients the way we all want to. And so what do I mean by Tomorrowland? Basically, that would mean a patient or family coming in to see us, getting a 100% accurate diagnosis that really informs their prognosis, and walking out with a treatment that we know will work. That's not the way it is now. Sometimes that happens at tertiary care centers like UCLA, but the hope is in the near future that will happen everywhere. So I like this picture because it illustrates that we are close enough to see our goal. We just have to figure out how to get that last mile. I don't have any financial disclosures. The learning objectives are here. Basically, I want to educate you about the types of genetic testing that are currently here and will be here in the near future. Understand why we would order genetic testing in psychiatric patients, and then talk a little bit about how model systems can be used to really help patients. And so that's what I'm going to talk about. First, I'm going to talk about genetic testing. Then I'm going to talk a little bit about what I've been doing for the past 4 or 5 years. And then finally, I'll talk about what I want to do in terms of using model systems to really pursue a translational approach. So this slide should come as no surprise to many of you, because Meredith actually showed something very similar 2 weeks ago. Many psychiatric illnesses are heritable, and the reason I show this slide is to illustrate, not that psychiatric illnesses are heritable, but to compare them to other illnesses that you see in medicine. So for example, here is breast cancer, here's Alzheimer's, here's type 2 diabetes, Crohn's disease, and what I want to convince you from this slide is that psychiatric illnesses, just as much, if not more, than other medical illnesses, are diseases in which knowing someone's genetic information can give us a great deal of information about what's going on in the patient and how we can help them. So currently, genetic testing is recommended for all patients with autism spectrum disorder and/or intellectual disability. I am very proud of the fact that here at UCLA, through ECPHP and now the CAN Clinic, all kids with autism are referred for genetic testing. And I know that many of you out, when you see kids in other settings, are also referring them, which I just say good job. The goal is for everyone out in the community as well to routinely get genetic testing on kids with autism. And so I'm going to keep evangelizing about this, and I'd like this to become one day to the point where, just like we ask uncomfortable questions of adolescents about sexual activity and substance abuse, every kid you see with autism, you ask, have they gotten genetic testing? It should just be part of the routine workup. Now, the first line testing that we get is called a chromosomal microarray, or array CGH, and then along with specific gene tests if there's a clinical suspicion. This testing, since about 2011, has been recommended by the American College of Medical Genetics, and through our experience and others' experience nationally, most insurance companies will now cover this as any other blood test. Now, if a chromosomal microarray is negative, and we'll talk about what that means in a minute, many centers, including UCLA, now offer whole exome sequencing. Whole exome sequencing is where you sequence just the parts of the genome that code for proteins, so it's about a third of the genome. And while CMA is covered by most insurance providers, coverage for exome sequencing is much more tenuous. And so as psychiatrists, we generally wouldn't order it. This is something that the geneticists would do. So if I'm going to ask you to ask every kid to get genetic testing, I should give you a rationale for why bother. Because getting blood from any kid is hard. Getting blood from an intellectually disabled or autistic child is even harder. So why would we do this? The main reason is to help distinguish between idiopathic and syndromic autism spectrum disorder. So idiopathic autism is where they have problems with social communication, cognitive rigidity, difficulties with language, but that's it. No other medical problems. Whereas syndromic autism is one in which they have the behavioral features of autism, but that is part of a larger medical syndrome. So they may have cardiac defects, birth defects, seizures. They may be predisposed to certain hormonal abnormalities or cancers. In many children, the behavioral features may be the first phenotype to manifest. So sometimes kids have obvious dysmorphology in these syndromes, sometimes they don't. And these kids may come to a developmental pediatrician or a psychiatrist first. Now, if you knew ahead of time that your child had a syndromic form of autism, then you may know to screen when this child is a young adult or teenager for some of the other medical complications that won't show up until then. And so this may help mitigate some of the medical components of these syndromes. And then finally, I don't want to understate the psychological benefit for parents. Many parents now that we've made a genetic diagnosis for have said, even though this doesn't affect treatment, even though the treatment for their child remains the same, simply being able to put a cause or a name to their child's particular condition is helpful because it links them to a larger community. So if you have Fragile X syndrome, there are now hundreds of families in the state who also have Fragile X syndrome that you're now a part of. So I'm going to talk briefly about what chromosomal microarray is. Again, Meredith gave a nice overview a couple weeks ago. But basically, you take a patient's DNA, label it with one color. You take reference DNA, label it in another, and you hybridize it, or you put it onto this chip with millions of, well, a million or so little spots. Each of those spots is a short DNA sequence that corresponds to a part of the genome. And you then shine a laser on this chip, and depending on the brightness of these spots, you can tell whether for that region of the genome there is more or less DNA than normal. And so if there's more DNA, that indicates a duplication. If there's less, then that would indicate a deletion. So the main thing that a chromosomal microarray finds are small breaks, small duplications or deletions in chromosomes. It won't find single gene mutations. It won't find triplet repeat expansions, for example. And so what does a negative test mean? This is true for a chromosomal microarray as well as for exome sequencing. A positive, when you send out for a test like this, you don't end up with a picture of a chip. You end up with a report. It looks something like a radiology report in which they say what this person's karyotype is, how many chromosomes they have, and then if there are any positive hits, they will outline what diseases those hits are associated with. So when you get a negative chromosomal microarray, it doesn't mean that the patient doesn't have a break, duplication, or deletion. It just means of the ones they found, none of them were associated with a specific illness. So a negative test just means that as far as we know right now, there isn't a disease-associated lesion. This makes a negative test a little bit difficult for parents to interpret because they think, OK, now what? This problem is even more pronounced for exome sequencing. Most of the variants are of unknown significance. So in the past couple of years, there was a large-scale exome sequencing effort done by on the Simon Simplex collection. We had a chance to actually look at some of that data, and only about 12% of the variants that they found in exome sequencing were known loss-of-function mutations. So that means in 88% of the cases, you had a mutation, but we just have no idea what it means. And so this really is the problem that I want to tackle. Right now, we have the ability to get a tremendous amount of genetic information from patients, but we're throwing most of it away because we don't know what it means. So as psychiatrists, we are standing right there. There is, in the very near future, there is going to be a flood of genetic data that will be coming to us from our patients. And patients will look to us to know what to do with this information. Right now, again, in our experience and in national published studies, about between 10 and 20% of cases of autism who have a chromosomal microarray have a positive test. That means the majority don't have a positive test. And so I think as a field, we need to get a lot better at being able to handle this kind of information and make something useful out of it. And so there is a plan in place for how to do this. This is not my idea at all. This was actually taken from MGH, and genetics labs across the country kind of use this paradigm for how things ought to work. So you start with patients and families. You have a particular illness, disorder, phenotype. You then start identifying underlying genes, risk genes. Next comes the step where you try to characterize a mechanism from the DNA to the phenotype, and this is sort of the black box. After you get past this step, you can do the really cool stuff in which you can start to look at targeted therapies, do drug trials, and then take it back to patients and families. Right now, through the DSM-5, we've gotten pretty good at identifying what a phenotype is. There's some, with RDocs, there's some debate about what we should actually call a phenotype. We're also really good at identifying underlying genes. In 2015, this is really factory science. There are huge teams of incredibly bright people, giant consortiums with thousands and thousands of patients, sequencers running 24 hours a day. So we have now, in the past year or so, really gotten good at identifying underlying genes. This step, though, is still a cottage industry. It's still individual people with their pet knockout mouse or other model system looking at one gene at a time. And so this orange step is really where I want to focus my efforts. We can talk about this later, but as an aside, in the case of Fragile X syndrome, we actually attempted to go all the way around this circle. And because of, and we had, people put in a lot of work in this step, but because of, in my opinion, flaws in this step, when we actually got to the clinical trials for drugs, they didn't pan out. And we can talk about that later if anybody's interested. So my research model is basically to take mutations found in patients with autism and introduce analogous mutations in the fruit fly, Drosophila. I then want to study the effects of these mutations on neural development. And it's a perfectly valid question to ask, what do flies have to do with mental illness at all? And the short answer is nothing. I am not particularly interested at all in the mental health of flies. What flies are a tool. They are a very regular and eminently studyable, if that's a word, nervous system, in which we know the precise steps of neural development down to the hour, and we can identify and isolate individual cells that are all at the same stage in development. So the tools available, the genetic tools available in flies, are really why I want to use this to study autism. So when I first started at UCLA, there was a lot of excitement about a gene called catnap2. It's a highly penetrant autism candidate gene. 70% of patients who have an early stop codon or a null mutation in in catnap2 end up having autism spectrum disorder. And so it was originally found in an Amish population where they noticed that people who were homozygous mutant for catnap2 ended up having a focal epilepsy, cortical dysplasia, and then a couple years later they noticed they all had autism, or many of them had autism too. Later, people went and decided to sequence this gene, specifically in about 3,000 cases of autism and 6,000 to 8,000 controls. And they ended up finding missense mutations that were found in patients with autism, and they never found those missense mutations in controls. Missense mutations, I should define what they are. As opposed to a mutation that causes a protein to stop early, or a protein or a frame shift mutation that completely changes the amino acid sequence of a protein. A missense mutation is where just one amino acid. What's the word? Say it again. Missense mutation. Yeah. So a missense mutation is where just one amino acid in the protein is changed. Many of these will probably do nothing. Some of them, though, will change the protein in ways that it's really hard to predict computationally. So that's why we thought, well, really the only way to figure out what these missense mutations do is to put them in a biological system. And I just wanted to highlight that in the case of CatNap2 and in the case of many other autism candidate genes, there is an incredibly high degree of evolutionary conservation between humans and flies. So each of these little shapes along the protein is a different protein domain. And I want to illustrate that in the human version and in the fly version, which is called Nexen 4, you have the exact same protein domains in the exact same order. So despite something like 100 million years of divergent evolution, this protein has stayed largely the same. The other important thing is many of these missense mutations were in amino acids that were the exact same throughout evolution. So that means that in the fly protein, I can make the change that was found in patients, and we can try to see how this missense mutation affects the overall protein function. So how do we go about doing this? The simplest way to go about doing this would have simply been to take a cDNA, which is just the protein coding sequence of this gene, make the mutation, and then overexpress it. The problem with that is you end up having what are called non-physiological levels of the protein. So you end up with 10 to 50 times as much protein as the cell would normally see. And so that becomes really difficult to translate that into what's happening in a patient. So instead, we decided to take the entire genomic locus, which has all the enhancer and promoter elements of this gene, put it in a bacterial artificial chromosome, or a BAC, and then modify it. And so we introduced the missense mutation, different missense mutations in the Nexen 4 locus. The other thing we introduced was a protein tag, which is a short peptide sequence that we have an antibody for that doesn't exist anywhere in the fly. And the reason we did this is so that we can follow our mutated protein in the cell and separate it out from normal wild-type protein. I want to point out that this type of approach can really only be done in the fly because the mouse and human version of this gene is about 2 million bases long, which is way too big to really handle. The fly version of this gene is only about 14,000, and so we can make these types of modifications. So the first thing we did after making flies that had mutant versions of Nexen 4 was to see, well, did these missense mutations somehow make the protein unstable? Did they make it degrade faster? And our answer is actually no, it did the opposite. So the mutant forms of protein were actually more stable than the wild-type version. So that would be a version with the tag but no mutation. Now, Nexen 4 homozygous mutants are dead. They die as early embryos. And so without certain genetic tools, it becomes very hard to study what does this protein do in an animal, because a dead animal that dies as an embryo just doesn't tell you very much. Luckily, in the fly, we have genetic tricks that allow us to have a completely heterozygous animal, but only have one tissue be homozygous mutant. And so this is a fly eye. This is actually what a normal wild-type fly eye looks like. It is made up of 800 individual repeating units in a very ordered crystalline structure. This is what a fly eye looks like when there is no Nexen 4 in that tissue. So the eye is there, but it's much smaller and very disorganized. Most of the cells involved are dead. These are eyes from flies in which, in the eye tissue, the normal Nexen 4 is gone, and only the autism-associated missense mutation forms are there. And so what I want you to notice about these eyes is they're not as bad as this, but they're also not wild type. So they have ommatidia, but the pattern is irregular. So from this data, we conclude that these missense mutations were partial loss of function alleles. And so this is actually the first time that anybody could show that these missense mutations were loss of function alleles. Whoops. There we go. I just want to orient you to what the fly retina looks like. So the fly retina is a sheet of neurons. On top of it are cone cells, which are glia, glial structures. Again, kids with CATNAP2 mutations don't have visual problems. I'm not particularly interested in the eye as a visual sensing organ, but this is a layer of neurons and glia. And we can study what happens in these neurons and glia to the missense mutated proteins. So in wild-type Nexen 4, all of the protein, which is shown in green, collects at the very top at these apical junctions of the photoreceptors of the neurons. And in a couple of the missense mutations, that's still true. But in 2 of the missense mutations, instead of collecting at the very top, it's now all over the neuron. It's all over the cell. So these 2 missense mutations, we could conclude, interfere with the correct trafficking of Nexen 4. So this is a blow-up of the cone cells, or the glial cells in the retina. And in the wild-type condition, Nexen 4 localizes to the junctions between cone cells and neurons, which forms this X pattern. In the 2 missense mutations that I showed you before, where they fill the cell, the junctions are still there, as shown by this other independent marker. But Nexen 4 with the missense mutation is no longer at these junctions. So in glial cells and in neurons, these missense mutations prevent Nexen 4 from getting to the right place. So finally, we wanted to see if there was any functional consequence of Nexen 4 not being at the right place in the cell at the right time. Nexen 4 forms a complex with other proteins at the cell surface. This is from work in mammals as well as in flies, one of which is contactin, another protein that is itself an autism candidate gene. And here we use a little genetic trick in the fly eye in which we can make patches of cells that don't have any Nexen 4, while the rest of the surrounding cells do. And so in these patches, indicated by the dotted lines, we see that when there's wild-type Nexen 4 being replacing the endogenous Nexen 4, the purple contactin staining looks just like outside the patches. There's no defect. But in the missense mutations in which the Nexen 4 is not localized to the cell surface, you also lose its binding partner, contactin. So we can say that the functional consequences of these missense mutations are that you don't have the Nexen 4 contactin complex at the cell surface, and that's likely what leads to all these problems. So what could we conclude about Nexen 4 from this? We showed that autism-associated missense mutations in CATNAP2 behave as recessive loss-of-function alleles. However, patients with autism are heterozygotes. So all those people that I told you about earlier, in which they sequenced them and found these missense mutations, they didn't have 2 bad copies of Nexen 4. They had 1 good copy and then 1 missense mutation. Now, when we made flies that had that exact same condition, they looked, as far as we can tell, normal. Didn't have a problem with eye development, could breed just fine. And so from this, we conclude that Nexen 4 mutations likely create a sensitized background in which other factors contribute to the development of autism spectrum disorder. And this leads to the question, well, what are these other factors? So I wanted to talk now about what I'm currently working on. And this is really an attempt to find out what some of those other factors are, not just for Nexen 4, but in general. I'm starting by working on a gene called CHD8. And CHD8 is the latest hot new autism candidate gene. It was found as a recurrent de novo mutation through these large sequencing efforts in simplex ASD. It is now the single most common de novo hit in simplex ASD. So there are something like 6 times as many cases of this gene being hit than any other gene that they found. And after sequencing around 15,000 control exomes, no known loss of function hit in CHD8 has ever been found. And that'll become important a little bit later. So what is CHD8? CHD8 is a chromatin helicase domain protein, and it leads to methylation of histones at specific sites. So basically, it regulates the expression of other genes. It represses some genes and activates others. And there's evidence that CHD8 represses beta-catenin and p53 signaling. And these are 2 pathways that are important in neural development, along with the development of many, many other tissues. just last year, people took neural progenitor cells derived from stem cells, human stem cells, and knocked down CHD8 in these cells in an attempt to find out, well, what genes are controlled by CHD8? The answer was something like 6,000 different genes. And that's fantastic from a scientific standpoint. From a clinical standpoint, that's not something you can do anything with. And so I want to show that just like catnap2, CHD8 is also highly conserved throughout evolution. So again, the boxes are different protein domains, and in the fly version of this gene, which is called Kismet, the same exact protein domains in the same exact order exist. Now this gene is very interesting to study in terms of finding out what those other factors are, because it is sort of a master regulator. It controls the expression of thousands of other genes. And so by studying this gene, we may be able to find out what some of those other genes are. Now, we've already started working on Kismet, and so we've been able to actually make a knockout of Kismet using a technique called CRISPR, which is a separate talk, but I'd be happy to talk about it later, in which we took out the first 1.8 kb of the gene. And we've been able to show that in the fly visual system, the system that we have the most tools to work in, Kismet is highly expressed during development in these neurons. Now, last year, there was a group at the University of Washington that published a really nice case series on CHD8 mutations. So they identified something like a total of 15 patients with CHD8 mutations. Some of these are deletions that take out the gene. Some of these are stop codons. Some of these are frame shifts. And they did some careful phenotyping on them. And the point of this paper, what they wanted to argue, was that CHD8 forms its own special kind of syndrome, much like CHD8-7 does with CHARGE syndrome. They wanted to argue that many of these kids have intellectual disability, have autism, have particular facial features like macrocephaly. But what really grabbed me about this chart was not so much what they had in common, but how they were different. So not every kid with a CHD8 mutation has autism. Not every kid has intellectual disability. Not even every kid. The only thing that's really, really consistent from their data, although it doesn't look like it from this chart, sleep problems are probably the most consistent feature of a CHD8 mutant phenotype. So I think when you study a single gene, many people naturally ask, well, OK, what is the phenotype? And what I'm interested in asking is, what is it about the genetic background that, given a mutation, makes these kids different? And the way that we want to try to answer this is by looking at combinations of genes. So when you sequence someone with, if you sequence anybody, if you sequence any of us, all of us are walking around with mutations. The average person has something like 50 or 60 mutations. I was rather shocked when I found this out, and I'm not sure if I entirely believe it, but most of us are actually probably walking around with a couple complete gene knockouts, and we're, as far as we can tell, fine. So I guess fine is a really broad term. So the question then becomes, how do you separate out mutations that lead to a specific disease versus mutations that just don't do anything? The way to do this, the way that I want to do this, is to test pairs of heterozygous mutations in the fly. So you would, when you sequence someone's exome, for example, in the papers that were published, they always talk about the de novo mutations, because that's what they have the statistical methods to make sense out of. But in addition to those random, spontaneous de novo mutations, every single one of those patients also had inherited mutations. And when we looked at about 25 of these patients, we found that each one of them had between 60 and 120 genes hit with an inherited mutation. And so it's not just this de novo mutation that is causing that person's phenotype. It's a combination of that de novo and inherited mutation. So for CHD8, we want to make flies that have one copy of CHD8 mutated, and then we will mutate these other genes that are found in common with patients with CHD8 mutations in order to find out which one of these interactions is important. And now for the translational part. So how can we actually go from the clinic to the flies? Recently, we're not quite ready to go live yet, so I'm not officially making a plug for the Developmental Neurogenetics Clinic, but we recently started a Developmental Neurogenetics Clinic in which we see children with known genetic disorders who also have psychiatric and neurological problems. In one of the first clinics that we had, we saw a 2-year-old boy with global developmental delay, hyperactivity, sleep disturbances, and maybe autism. This child's actually in the foster care system, and getting him fully tested for autism is problematic, but hopefully we'll have it done in the near future. So he has an inherited missense mutation in CHD8. And remember when I told you before that they sequenced 15,000 people and never found a hit in CHD8. That's because the way they defined hit was a loss of function mutation. So missense mutations in CHD8 clearly do exist. And through this clinic, I was lucky enough to be able to interview the mom from whom he inherited this CHD8 mutation. Now the mom, she had her child taken away by DCFS. She is on antidepressants. She has mental health issues, but she does not have global developmental delay or autism or intellectual disability. And so that raises the question, what is this missense mutation doing in this child? The other really interesting thing about this child is this is not the only gene in which we found a missense mutation. There's a transcription factor called RFX3 in which this child also has a missense mutation. And I'm calling it de novo only because we could never get DNA from the father. So it might have been inherited from the father, but we just have no idea. Now, both of these mutations are actually in conserved amino acids in the fly versions of these proteins. So right now, we are making a fly to test the hypothesis that these mutations are causal in patients. And so this is how I'd like to actually use flies to inform patient care in this clinic. Because for the first time, rather than saying, well, we have genetic information that is suggestive, we can now make a statement that's saying, no, these two mutations really are causing the child's mental illness. But it actually goes beyond that. So the Developmental Neurogenetics Clinic is in its infancy, but at some point, I think actually psychiatry can inform genetics as much as genetics informs psychiatry. What we are really good at in psychiatry and psychology is phenotyping people. And by phenotyping collections of people with the same mutations, we will finally be able to answer the question, how much of the salient features of this patient are actually explained by these genetic lesions? I showed you that chart before in which patients with CHD8 mutations had a whole range of phenotypes. Some of them had it, some of them didn't. But the question is, of those features, how much of that was due specifically to CHD8, and how much of it was other factors, environmental factors, other genetic background mutations? And so that, I think, is a major question that needs to be answered, and hopefully flies provide a tractable organism in which we can start to address that. So I'd like to conclude by saying that genetic testing is becoming routine in psychiatric patients, whether we want it to or not. But there is a gap in our ability to interpret the data. Drosophila is a powerful tool to model combinations of mutations found in these patients. And the goal is that we need, as physicians, to be better able to interpret the genetic findings in these patients. So I'd like to thank Simon's Foundation, who funded most of this work. I'd also like to thank the ACAP for also partially funding this work through a pilot award, UCLA. I'd also like to thank my mentor, Larry Zapersky, and Poonam Patel and Kartik Bapu, who are members of the lab who helped me on this. Members of the D&G clinic, Shafali Jesti and Julian Martinez, who are the other clinicians, and Carrie Bearden as well. And finally, the members of the CAN clinic, Jim and Amanda. And that's it. Is there any questions?